So I'd like to begin by thanking the organisers for kindly inviting me. The talk is going to be in three parts. I'm taking my cue actually from the last speaker, from Dr. Dan Peterson's excellent lecture, in which he alluded to the fact that there may be merit in carrying out magnetic resonance spectroscopy scanning of the brain. And indeed the first two parts of my talk will be about MRS of the brain. The first will be a recap of previous results, and you'll see that they are germane to what has been described this morning in terms of infections. And the second is a very recent magnetic resonance spectroscopy study which I've carried out at a higher field strength, in fact the highest field strength that has ever been used, I think, in myalgic encephalomyelitis patients. And then the third and last part of my talk will be a pilot structural MRI study of severe ME. So starting then with a recap of our previous 1.5T MRS studies, there have been three studies really looking at proton spectroscopy of the brain and examining in particular the choline levels. All the studies I'm going to describe have taken place here at the Hammersmith Hospital. And this is one of my colleagues, Dr. Ian Tresden, who was here last night and is actually the most senior fellow of the Royal College of Psychiatrists, who has been supportive of the biological approach that my research is taking into this disease. So both our study and the study by Professor Abhijit Chowdhury were systematic comparisons of the brain in terms of proton spectroscopy at 1.5 T between ME patients and age and gender matched controls. And we both found an elevated level of free choline, so this peak here. And then he pointed out to me that Tomoda and colleagues had published a non-systematic case series of 3 juvenile sisters with myalgic encephalomyelitis who also showed increased levels of choline, but that was compared with a database of non-matched controls. And it's relevant to what's been described today, this morning, and also last night in Hilary Johnston's excellent lecture on her book Osler's Web in respect of an infectious etiology. You'll know that our outer cell membranes are made of lipid bilayers, as are the membranes of many intracellular organelles, such as the nucleus and the mitochondrion. And the polar head group there, these things, are often choline. And it has occurred to some of us that if one suffers from certain infections, then certain organisms are able to prevent our cells from biosynthesizing the long-chain polyunsaturated fatty acid that is supposed to be present at the SN2 position of the phospholipid. This is just a Fischer projection of a phospholipid molecule, so that fatty acid there. And therefore, one can't produce sufficient quantities of these phospholipids, and therefore the level of this choline, the free choline, would rise. And it's the free choline here, rather than the bound, phospholipid-bound choline that's been measured with 1.5T MRS. So 3 studies, and they all converge to the same results. So now I'm going to move on to the second part of my talk, which is using proton spectroscopy, this time at a higher magnetic field strength of 3 Teslas. You'll be aware of the preliminary evidence suggesting that there is increased oxidative stress in the body in myalgic encephalomyelitis, and the question is to what extent this manifests itself in the brain itself as well as peripherally. And a lot of work, a lot of hypotheses have suggested that glutathione, reduced glutathione, that's GSH, in particular, might be reduced in level. On the other hand, a study like this study from Australia found that there was actually no significant difference in reduced glutathione, GSH, levels in the erythrocyte cell membrane between, or erythrocytes, between patients with CFS and age and gender-matched controls. But nevertheless, the hypothesis still continues to be put forward that if we could find reduced GSH, then this would, in particular for the brain, be associated with increased blood-brain barrier permeability. So that's certainly the conclusion from some infrahuman experiments. But this is hypothetical. It's a hypothesis paper from Canada published back in 2001. Glutathione is an interesting molecule. It's a thiol-containing tripeptide consisting of glycine, cysteine, and of course glutamic acid or glutamate. And here you see it in its reduced form, interacting with hydrogen peroxide and forming water. This reaction is catalyzed by the glutathione peroxidases group of enzymes, which are widely distributed in the body. They are mostly specific for reduced GSH as a hydrogen donor, but they can act on other peroxides. For example, they can catalyze the GSH-dependent reduction of fatty acid hydrogen peroxides, and the peroxide group itself is then reduced to an alcohol, this thing here, NOH. The great thing about proton neurospectroscopy at 3T is that instead of just those 3 peaks I showed you earlier, choline, creatine, and acetyl aspartate, we can now open up the field and see a large number of peaks. And in this particular case, the peaks have been fitted mathematically to in a way that allows one to work out the levels of glutathione, GSH. So this is exactly what I did, really. And I used 64 signal averages, and I was careful to optimize the magnetic field homogeneity, both globally and in terms of the voxels I was looking at. And here's the answer. The units are arbitrary, by the way. While the mean value of glutathione seems to be lower than that in the age and gender-matched controls, 2.7 versus 5.2. In fact, taking into account the higher variance, the standard deviation of the control data, in fact, there was no significant difference. And indeed, I used an equal variances assumption, but even if you don't, the data still come out with a non-significant result. So that's consistent with the erythrocyte reduced GSH data results from Australia that I showed you earlier. So finally, I'm just going to talk briefly about the structural pilot structural MRI study, also at field strength of 3T, and this is essentially in severe ME patients. I decided to use voxel-based morphometry because it's really a very fair way of assessing whether or not there are changes between voxels between the two groups. I mean, it's rather important because a lot of this is almost a single-handed study, really, these days. And, of course, I know who belongs to which group. I'm totally unblinded in that respect, so I had to find a method that was particularly operator-independent, if you like. The groups, I dichotomized them to 2 age and gender and ethnicity-matched groups, and I used my initial T1-weighted sagittal MRI scans, the scans I acquired early on at each protocol from the left ear, going that way to the right ear. Just for those of you who are not totally familiar with MRI, so these are pixels. This letter E is made up of lots of little oblongs which are filled in. Those are individual pixels, and so what I'm looking at are 3-dimensional versions of those, in other words, voxels. As I say, the method that I've chosen is non-operator dependent, and I can feed in absolutely no a priori information. All I tell the workstation as I'm programming it is that this is the list of the first set of subjects belong to one group, and the next lot of subjects I'm feeding in belong to the other group. That's all. I don't bias it in any way at all. The data, the images, have to be transformed onto a standard space, and non-linear transformation is the method of choice really these days. So construct an MRI template, produce a suitable statistical design, and of course I have to remove the bits of the brain, the head that don't matter, such as the skull and the hair and things, just the brain that matters, of course. So I have a choice as to what form of registration to use. Registration is when you have to map one set of data to another accurately. And I actually go for affine registrations. I think that's a very fair and appropriate way of doing it. And this is the definition of an affine space that I use, which any of you who are familiar with applicable differential geometry will recognize as being a very good model of what happens in reality. And I also choose to use a cluster-based correction for multiple comparisons, because clearly you're comparing all these individual voxels with voxels from another group, and so I have to correct for that, and therefore set my P thresholds vanishingly low. And I don't even finish there. I then randomize the whole thing with 5,000 permutations. I won't go through the statistical details with you unless you want me to, but it's highly, highly robust. These are the results. So these are coronal images, so looking at the brain like that. These are axial or transverse images looking at it from above or below. It's essentially the same thing. And these are sagittal ones looking at it from the side, from the left side. In fact, no, looking from the right side. So the areas that I've coloured in a sort of fluorescent green are those areas where the grey matter was less dense in the ME patients compared with the age and gender matched controls. And the actual images of the brains are just images I've superimposed as a template, which are from the MNI 152, the Montreal Neurological Institute's 152 brain database. And here's another one looking at a different level, because I can't show you the 3D brain that you can rotate around, so you have to take different sections. The other bit of bias that could have been introduced is my interpretation of what these bits are. And I could have said, well, I think that's cingulate cortex, but maybe I have some bias for that. I mean, I'd have had a bias for looking at the brainstem because of the chairman's seminal paper back in the 1990s showing reduced blood flow in the brainstem in CFS. So I thought I must remove even that bias as well. So I chose, instead of using my knowledge of anatomy, I chose instead to use, well, I did use, of course, I corroborated everything using a Harvard University, Harvard University, Oxford University joint atlas, and checked everything against the electronic atlas. So this is a summary of the brain changes in severe ME. The parts of the brain that show reductions in grey matter density are the right inferior frontal gyrus, both the pars apicularis and the pars triangularis. I'll show you these in a moment. I've tried to find some pictures on the internet yesterday to illustrate some of these. The Singlet Gyrus, particularly the anterior division thereof, the left precentral gyrus, the left middle frontal gyrus, and, as you saw earlier, the cerebellum. I couldn't find a right inferior frontal gyrus, so this is the left, and you can see it there. There's the inferior frontal gyrus, and there are the two parts that I showed you as being identifying as having reduced values. Now, in terms of right inferior frontal gyrus function, there is good evidence that, in fact, and these are all just hypotheses as to what the functions are of these parts of the brain. There is evidence that this part of the brain, the right inferior frontal gyrus, is involved in the mismatched negativity that's associated with the auditory evoked potentials. And there's good evidence from FMR studies, actually. So I think it's something we just bear in mind as a possibility. Now, in respect to the Singlet Gyrus, well, first of all, it's the Singlet Gyrus here in this awful colour, and you're probably aware that it has numerous inputs from other parts of the brain, such as the anterior thalamus, etc. One of the key things about Singlet is that it's involved in executive control to suppress inappropriate unconscious priming. And one of the best ways of testing is to use the Stroop Test. So the Stroop Test preferentially activates anterior Singlet Gyrus amongst other regions of the brain. For those of you who are not familiar with the Stroop Test, you have a set of names of colours that are printed essentially not in their own colour, and then you have to try to articulate the colours they're printed in and ignore the colours that are written. And so, in essence, you're getting two bits of information coming in, two important parts, the name of the colour and the colour it's actually printed in. And then the anterior cingulate, amongst other parts of the brain, has an executive function in that it has to take both and discard the unwanted bit of datum, or data, the name of the colour, and let through the actual colour that the word is printed in, and that then is those data are then fed through, presumably, to Broca's area and allow one to articulate the correct answer. Except I have colleagues who can't get this right, actually. Probably a job waiting for them at the CDC. No, sorry, I shouldn't say that. It's being recorded. The left precentral gyrus, that was a big surprise to me because, of course, the left prefrontal gyrus is Brodmann area 4. It's the left primary cortex, and it controls the contralateral part of the body. And this is just a picture of the motor homunculus that Wilder Penfield first, the great neurosurgeon from Canada, first talked about back in the 1950s. Left middle frontal gyrus, that was easy to find, and there it is, there. But in terms of its functions, not absolutely sure. That there seems to be some good evidence that malfunction of the middle frontal gyrus would impair one's ability correctly to write Mandarin. So, and the cerebellum has, it's down there, has many, many functions. Some people think it still is only related to sensory perception, coordination and motor control, but I'm absolutely sure that it has functions in respect of attention and the processing of language, music, and other sensory temporal stimuli. So, in conclusion, I think we've got clear biochemical evidence of biochemical and structural brain changes in ME. These do strongly favour the notion of a biological basis to this neurological disease. And I just want to acknowledge various groups, ME Solutions, Merrick, the MRC, several MD students, particularly Maged Agur, the many, many ME support groups who've helped with this study, and most of all, the ME patients themselves who have made the long trek to the Hammersmith and kindly volunteered to take part. Thank you.
Fascinating talk. What questions? Lady here.
Thank you. That was incredible. Do you think that the concept of neuroplasticity and the ability of some parts of the brain zones to take over functions that are lost offers some hope for cognitive dysfunction, and also other supplements such as phosphatidylserine and things, lecithin, to build up the phospholipid membranes and such?
The straight answer to the question is yes and yes. I think that it is amazing, the plasticity in the brain, in the adult human brain is an incredible phenomenon. Not many people know, but I was actually the first to describe the ability of the adult brain to regenerate. It's a published paper back in 1999. I think, in 1998. But most of my colleagues, in their infinite wisdom, or almost infinite wisdom, were able to point to various textbooks and say that this must clearly be wrong, because the textbooks say that the adult brain doesn't regrow. I should have pointed them to my own textbooks and said, look, it's going to change. But in the last 3 or 4 years, researchers in America working with the adult mouse, the murine brain, have shown that the mouse brain can also regenerate, and therefore we can now accept that the adult human brain can also, because you have to show it in a mouse first, you can't show it in humans. You mentioned phospholipid membranes. Yep, we did some work, which I think I published in November last year, on Huntington's chorea, which is, of course, a devastating illness. What I found there was that treatment with a particular type of long-chain fatty acid, EPA, was not only associated with an improvement clinically, but also with just that very phenomenon, regeneration of the brain, and not just any old part of the brain, but in fact, interestingly, the anterior part of the caudate nucleus and part of the thalamus, and that's interesting because the caudate nucleus is one of the first parts of the brain to presumably be affected by the Huntingtin protein and to become abnormal.
In the very severe group whose sleep is very badly affected, what do you find on a SPECT scan?
I missed the very beginning of your question, sorry.
What's the SPECT scan like? What's the SPECT scan like in severely affected?
But those in particular who've got very bad sleep patterns, I just wondered what that showed.
Well, I think perhaps you might be a better person to answer that now. I've carried out SPECT scanning in certain groups, and not in ME yet. I mean, I'd like to, but I haven't got the funding to, so I don't know the answer, I'm afraid.
Thanks very much, Basant. That was terrific. Are you associating the increased choline levels with some indication of a viral infection? And it seems from what you were saying, if I understood it correctly, that the problem isn't the choline, the problem is the fatty acids not being there, so that you're losing those, presumably by an oxidative process, I don't know, but have you any comment on that? And what do you make of the data from Van Spence, which showed actually very clear changes in glutathione levels and also corresponding changes in oxidative LDL levels, which really are not the same as not the same as what you've been saying. That the glutathione levels did in fact go down, and the oxidative LDL levels went up. I just wondered if you had any way you could rationalise or square that circle, really.
Okay, so there's two sets of questions there, I think. In respect of your first set of questions, I mean, that's my working hypothesis. I could be wrong, and there may be some other explanation for the increased choline. Maybe something to do with acetylcholine, for example, being broken down, but I'm not sure I really buy that, but it is I can't rule that out. My gut feeling is that this is choline that really should be incorporated as polar head groups. So what you said is what I believe, I think, until I'm shown to be wrong, of course, as with all science. In respect for your second question, I mean, I wasn't sure that Van Spence had actually demonstrated reduced levels of reduced GSH in the brain itself.
That's true. It wasn't in the brain. He was measuring it in circulating blood, really.
But you make an excellent point, which is that there are findings in the periphery, as it were, outside of the brain. And given that so many of the symptoms and signs of myalgic encephalomyelitis are related, one presumes, to the brain itself, it does seem to be that one does need to use the best technology we have directly but non-invasively to look at, for example, glutathione in the brain itself. And having done that, I can't find a difference, so I can't square that circle.